IITA Bioscience Center. This is one of the major research labs in IITA. Here we use molecular tools to assist the conventional breeders. And also we are involved in training and capacity building of national researchers. We offer laboratory bench spaces to students from different universities, and we host a lot of graduate students. Right now, I'll be taking you around to see some of the major instruments we use in analyzing our data. Thank you. Let's see. This is DNA extraction lab. This is where we extract genomic DNA from plant tissues. We have quite a number of instruments in this room. We have the Fume hood, whereby when we are working with hazardous chemicals, it has to be done under the Fume hood. We have the water bath, where we incubate. We have the centrifuges. These are the centrifuges that we use in the DNA extraction lab. We also have the RNA Isolation lab. That's where we extract RNA from plant tissues. The lab also has almost the same instrument we have in DNA extraction lab. The major difference is RNA and DNA, they are both nucleic acid, and while doing extraction, we need to do them separately in order to avoid cross-contamination. Let's go. The room we have here is the instrument lab, whereby we have quite a number of machines, such as Lyophilizers that we use in fish drying samples. We have genome grinder that we use in grinding samples. It's just an automated machine that grinds about 384 samples in less than 5 minutes. We also have centrifuge and shaker. This is ploidy room. This is where we run ploidy analysis. We have a machine known as flow cytometer. We use the equipment to determine the ploidy level of plants. Majorly, we use the equipment for clonally propagated plants such as cassava and yam, even plantain. Let's go. This is real-time PCR lab. Here we have two types of PCR machines. Real-time PCR machines, we have 7500, then we have Roche light cycler. This Roche light cycler performs two major analysis. We use it for gene expression studies, then we also use it for CASP genotyping. Also, we have another instrument, flow star plate reader. We use it for quantification of nucleic acid. The machine can quantify up to 384 samples in less than 10 minutes. We also have another nanodrop spectrophotometer. We also use this instrument to quantify nucleic acid and proteins. It can also quantify up to 96 samples in less than 5 minutes. Let's go to the next one. Here is our sequencing room, whereby we have genetic analyzers. These genetic analyzers are used for both sequencing, which is to determine the order of nucleotide bases, as well as for capillary electrophoresis. This is just analyzing DNA fragments. We have other methods used in analyzing DNA fragments. Reaction laboratory. We have quite a number of PCR machines here, which are used for the same function. We use the instrument to amplify a fragment of DNA to generate several copies of that particular DNA sequence. This is the Nanodrop Lab. Here we have Nanodrop spectrophotometer. We use the instrument for quantification of nucleic acid and proteins. We also have some other equipment in this room like FTIR. Then we have pipette maps. That's a robotic machine for pipetting. Here is Igaro's Gel Electrophoresis Laboratory. We have different kind of electrophoresis. Agarose gel is one of the methods we use in analyzing DNA fragments. We have PAGE, which is polyacrylamide gel electrophoresis. We have capillary electrophoresis. So here we make use of agarose to make DNA, to visualize DNA. We have two DNA types. We have the PCR amplicons, then we have the DNA, genomic DNA. So we have here the power pack. That's where we load our DNA and we run it using the power pack. And after about 40 minutes, we can visualize DNA under UV transilluminator. So that's all about the tour of Bioscience Center of the International Institute of Tropical Agriculture. I hope you enjoyed the tour. Thank you.